So I was going through our notes for this deep dive, and one name just kept jumping out at me. Vampyroteuthis infernalis.
Quite a name, isn't it?
It is. I mean, they call it the vampire squid from hell. It sounds like something from a monster movie.
It really does, a creature of myth. But this deep-sea enigma, it's very real. And it's quietly rewriting our whole understanding of cephalopod evolution.
Absolutely. And it lives in this realm of just perpetual darkness, hundreds, sometimes thousands of meters down.
It's a phylogenetic relict, a real survivor. People often get it wrong. It's not a true squid, not a true octopus.
It's the sole remaining member of its ancient order, the last one, dating back over 183 million years.
And that's what makes this so exciting. This is the big news. Scientists have finally sequenced its genome.
And what they found was it was a shock.
Astonishing. It's the largest cephalopod genome ever sequenced, up to 14 gigabases.
That's a massive instruction manual for such a small, reclusive animal. So our mission today is to dive into that blueprint. We want to unpack why this genome is being called the Rosetta Stone for cephalopod evolution.
It gives us this direct look, for the first time, at the shared ancestor of modern octopuses and squids from nearly 300 million years ago.
And it might just reveal some of the secrets behind how intelligence evolves in the ocean. Okay, so before we get into the genetics, I think we really need to appreciate just how tough this animal is.
Oh, for sure. The vampire squid from hell, it makes its home in one of the ocean's absolute most unforgiving environments.
It's the bathypelagic zone, right? Specifically something called the oxygen minimum zone, or OMZ.
Exactly. We're talking 600 to 900 meters deep, sometimes more. And to give you an idea of how deadly that is, oxygen saturation there can drop to as low as 3%.
3%. That's a vast toxic expanse that most complex life just avoids.
They can't respire. Most cephalopods, which are incredibly active, would suffocate almost instantly.
So how does the vampire squid do it? How does it survive, let alone complete its life cycle where every breath is a struggle?
It's basically solved the problem of energy scarcity. For starters, it has the lowest mass-specific metabolic rate of any known cephalopod.
It's built for efficiency.
Total efficiency. It's designed to operate on an absolute minimum of energy.
It's the deep-sea champion of just low power mode.
That's a great way to put it. And the engineering goes even deeper. Its blood, which is blue, uses a protein called hemocyanin.
Right.
And that hemocyanin is chemically engineered for an extremely high oxygen affinity. So it can just scavenge and bind to every last molecule of O2 it can find.
And I imagine it doesn't waste any of that precious oxygen.
Right.
Its body composition is key here.
Crucially, yes. It achieves neutral buoyancy passively. Its tissues are full of ammonium, giving it this gelatinous quality that matches the seawater's density.
So it doesn't need to burn energy constantly swimming just to stay in place.
It just drifts.
Which, again, that really goes against the ferocious name, doesn't it? If it's running on fumes and just drifting, how on earth does it eat? Does it attack things?
And that's the wonderful contrast. The name suggests this fierce predator, but biologically, it's a sedentary detritivore.
Oh, what?
The detritivore. It uses two long retractile filaments, they're actually modified super thin arms, to just passively collect marine snow.
Ah, marine snow. So all the gunk-dead organisms, fecal pellets, zooplankton remains drifting down from the surface.
Precisely. It's dining on dust. The animal might spend, 90% of its life just hanging there, almost motionless, waiting for dinner to literally fall into its arms.
That strategy makes perfect sense for the OMZ. It's all about conserving energy.
Exactly. Its entire existence is a masterclass in minimal energy expenditure.
But even with this passive diet, it has some incredible features for defense. The eyes, for instance, are huge.
Proportionally the largest in the entire animal kingdom. Yeah. And they're not designed for sharp vision, but for extreme sensitivity to light. They can spot the faintest flicker of bioluminescence in the dark.
And if it's threatened, I read about its main defense, this pumpkin or pineapple posture.
Right. It curls its 8-webbed arms outward and backward over its body.
That must look pretty intimidating.
It makes it look much bigger, and it exposes these fleshy spine-like structures called cirri. It creates this dark, forbidding silhouette. But the real show is its last resort.
The famous burglar alarm.
That's the one. If it's really agitated, it ejects a cloud of sticky, bioluminescent mucus from its arm tips.
So it's not like ink.
No, it's this glowing, sticky cloud. And it doesn't harm the predator, but it sticks to it, essentially lighting the attacker up in the dark.
Which then attracts bigger predators.
Exactly. The attacker becomes the target, and in all the confusion, the vampire squid just slips away. It's a complex, high-energy defense which shows that even with a minimalist lifestyle, evolution can cook up some spectacular tricks.
Okay, now let's get to the core of this discovery, the genome. You mentioned the shock factor of its size, 11 to 14 gigabases.
It's just enormous.
How does that compare if we put it next to, say, a common cuttlefish?
The cuttlefish is about 5.5 GBP, or the California two-spot octopus, which is one of the most studied, is only about 2.2 GBP.
So the vampire squid's genome is several times larger than its modern, highly intelligent relatives.
It is genuinely perplexing on the surface.
Yeah.
How can the genome of this sluggish, ancient, passive creature be so much bigger?
And we know from biology that a bigger genome doesn't mean a more complex organism.
Right. If it did, the common onion with its 16 GBP genome would be a genius.
So what's going on here?
It's a case of genomic inflation. The sheer bulk of the vampire squid's DNA, a staggering 62% of it, is made up of repetitive elements.
What was once called junk DNA.
Exactly. These are non-coding sequences, things like signs and lines that just repeat over and over.
So it's like a computer hard drive that's been running for hundreds of millions of years, but it never deleted the temporary files or defragmented. It just keeps copying and pasting huge stretches of code without adding new programs.
That's a perfect analogy. It suggests that the vampire squid lineage has mechanisms that just let these repetitive bits accumulate over time without cleaning them up.
Whereas modern cephalopods have better genetic housekeeping.
Much more robust systems, yeah. They seem to keep their genomes compact and streamlined. The vampire squid, it seems, tolerated the mess in exchange for genomic stability.
Which makes for an enormous, chaotic data set. I mean, analyzing a genome with over 11 billion base pairs, especially one that's so repetitive, must be an astronomical computational challenge.
Oh, it is. Finding the needle of actual evolutionary innovation in a haystack that big requires incredibly specialized tools, sophisticated automation, advanced software.
And that challenge is one that, many organizations face today, which is why we're proud to be sponsored by Embersilk.
It's a very natural fit.
This deep dive is sponsored by Embersilk. If this conversation about sequencing 11 billion base pairs of messy, chaotic biological data has you thinking about your own organization's data challenges or how to implement complex systems efficiently, you really need to talk to Embersilk. Whether you're struggling with AI training, optimizing automation pipelines, or integrating new software, Embersilk specializes in taming that kind of chaos. They help you uncover exactly where intelligent agents could make the most profound positive impact for your business or your personal life.
Turning massive data sets into actionable knowledge.
Exactly. So if you're looking for solutions to manage your complex AI needs and streamline your systems, stop sifting through the junk data by hand. Head over to embersilk.com today and see how their solutions can help you. Welcome back. We've established that the vampire squid genome is enormous and full of these repetitive elements, but why is the specific architecture the key? The Rosetta Stone to understanding the whole cephalopod family tree.
Well, what's fascinating is what the structure itself reveals about that divergence point about 300 million years ago. Taxonomically, the vampire squid is an octopodiform.
So it's in the octopus group, the 8-armed group.
Right. But what the researchers found was that its genome, its chromosomes, retains an ancient 10-armed or squid-like chromosomal architecture.
Wait, hold on. So this 8-armed creature has the genome layout of a 10-armed creature. That seems very counterintuitive.
It is. And that's the key. It implies that the common ancestor of all modern cephalopods, both the 8-armed octopuses and the 10-armed squids and cuttlefish, had this specific layout.
And the vampire squid's lineage just held on to it.
It just retained that ancient, stable structure, even as it was accumulating all that repetitive DNA.
So the big evolutionary revolution happened after the octopus lineage split off.
Precisely. The incredible intelligence and adaptability we see in modern octopuses were achieved through an early, rapid phase of major chromosomal mixing and fusion. This massive reorganization was an irreversible process.
Which helped drive their specialized adaptations.
Yes, but it meant they sacrificed that ancestral blueprint. The vampire squid is the genomic living fossil because it preserved the original layout. It lets us reconstruct the evolutionary starting line.
And this lines up beautifully with recent fossil discoveries, doesn't it? The 330-million-year-old fossil, Cephalopod bedeni, found in Montana.
The oldest known ancestor of octopuses, yes.
And it had 10 limbs.
10 limbs. It directly supports the conclusion from the vampire squid's genome. The whole group evolved from a 10-limbed, squid-like ancestor. The fossil record showed us the physical body, and now the DNA gives us the genetic instruction manual for that same ancient body.
It's a perfect alignment of paleontology and genetics.
Mhm.
So cephalopod evolution produced some of the most complex and frankly intelligent creatures on Earth, right up there with vertebrates in many ways. But they didn't do it through whole genome duplication, which is how vertebrates got a big boost.
Right, they didn't just copy the entire instruction manual, they got creative. They achieved their complexity by selectively expanding and reorganizing specific key gene families and by developing some very unique post-transcriptional tricks.
Let's talk about one of the key examples, protocatecherins. These are cell adhesion molecules, right?
Basically, yeah. They're like the molecular glue and signal posts that determine how neurons connect to each other, how you wire a complex brain.
And the expansion of these genes in cephalopods is spectacular.
It's off the charts. Most other mollusks, like snails and clams, they have maybe 17 to 25 of these genes. Even mammals, with our complex brains, we only have around 50.
But the California two-spot octopus?
Octopus bimaculoids, yeah.
Yeah.
It has a staggering 168 protocatecherins.
168. That's more than triple what a human has. So what does that explosion of genetic tools actually let an octopus brain do?
It facilitates this vastly complex network of connections, especially in their decentralized nervous system, where 2/3 of the neurons are actually in the arms.
So it's not just a big brain, it's an incredibly finely wired brain.
Exactly. That massive number of protocatecherins allows for these really precise, distinct connections between neurons, which enables advanced stuff like complex learning, problem solving, and that incredible camouflage control.
Which requires massive processing power across its whole body.
It's a textbook case of convergent evolution. They arrived at a complex nervous system, but on a totally different path from vertebrates.
And beyond just expanding their gene toolkit, they also found a unique way to edit the code after it's been copied. Tell us about A to I RNA editing.
This is truly a marvel of evolution. A to I RNA editing is really common in their nervous tissues, and it lets them modify their genetic messages on the fly.
So the DNA is the blueprint, but they can make little edits to the photocopies before sending them to the factory floor.
That's a great way to put it. One single gene sequence in the DNA can end up producing a whole diversity of different proteins.
So one gene can act like 10 different genes in the brain.
Precisely. It's a massive fine-tuning mechanism. If the environment suddenly changes, say, the water temperature, they don't have to wait for a slow genetic mutation. They can rapidly edit the proteins to meet new demands.
Which gives them that incredible real-time adaptability we see.
In their behavior, their camouflage, everything. And this all seems to trace back to that ancient split. The reorganization of genes we see when we compare the vampire squid to the octopus. That seems to be the launch pad for these immense innovations.
So the intelligence of the modern octopus wasn't built on a whole new set of genes.
But on an unprecedented reorganization and a really creative use of the ones they already had.
So we started this deep dive with the vampire squid from hell and realized it's less of a monster and more of a meticulous deep-sea scavenger.
A very patient one.
But by sequencing its huge, strange genome, scientists haven't just figured out one animal. They've built a complete framework for understanding the evolution of the entire highly sophisticated cephalopod class.
It's a testament to the immense value that's hidden in life's most ancient remnants. The vampire squid's stability preserved that ancestral blueprint, which lets us measure the incredible leaks taken by its cousins.
And looking at that genome, with 62% of it being so-called junk, it's easy to see it as inefficient. But you're suggesting we should see these chaotic genomes not as waste, but as reservoirs of potential.
Absolutely. The constant dynamic genetic restructuring, the tolerance for these massive repetitive elements, it showcases evolution's powerful, opportunistic capacity for innovation. It reminds us that often the largest, messiest data sets in biology aren't dead ends.
They're adaptive playgrounds.
Exactly. They're poised, they're full of unexplored potential, just ready for future unexpected evolutionary leaps. And I think recognizing that potential, whether it's in a deep-sea creature's DNA or in our own technology, that really highlights the ongoing, hopeful progress of life itself.
That is a brilliant thought to end on. If you enjoyed this deep dive into the genetic secrets of the abyss, please subscribe to the show. Hey, leave us a 5-star review if you can. It really does help get the word out. Thanks for tuning in.